Let's begin with a new chapter today, and that is genetics. So, first of all, let's try to understand what exactly is genetics. Genetics is called the science of heredity and variation. We all know that we resemble our parents in one way or the other. So, what resemblance we bear to our parents is due to heredity. That is what we have inherited from them. And what own features we develop is due to variation. So, what exactly is genetics? The study of heredity, that is, transmission of bodily features, both similarities and differences. Differences would be the variation from parents to offspring. And we also study about the laws which are relating to transmission. So, this is the definition of genetics. What exactly is heredity? The term heredity is defined as transmission of genetically based characters from parents to offspring. So, the characters which are transmitted from parents to offspring we study in heredity, and the whole science which talks about heredity, that is similarities, and even variation, that is differences, is known as genetics. So genetics is the broader science. It includes heredity. Now, what features do we inherit from our parents? They may be color of the eye, hair texture, you can say, that is curly or straight, eyebrows, heavy, bushy or thin, hair on the joint of middle fingers, whether they are present or not, color vision, that is whether you have normal vision or you have color blindness, tongue rollers, whether you are able to roll your tongue in a U-shaped tube or not, whether you are right-handed or left-handed, whether you have normal skin coloration or you have inherited albinism from your parents, whether your ear lobes are free at the base or are they attached to your main head area, whether your lips are thick or thin, whether your blood group is Rh positive or Rh negative. These are some of the examples of the characters which we inherit from our parents. And this inheritance is studied under heredity, and the science of heredity plus variation is studied under genetics. Now, what is variation? Human beings as a species share many characters or traits among themselves which identify with the species. Yet, various races or tribes look different in several features. Even within the same race or tribe, the individual members of the population show differences. So, these small differences among the individuals of the same species are called variations. So, how you resemble your parents we study under heredity, and how you differ from them and develop your own features you study under variations. Like you've studied that in meiosis crossing over occurs and the gametes formed are haploid but they are crossovers. So something new happened. At chiasma, exchange of genetic material occurs. Something new happens. A combination is obtained. Neither it's purely maternal nor purely paternal. And that is why there is a variation in the offsprings as well. So learn the definition of genetics, heredity and variation, and some examples of the characteristics which or characters that we inherit from the parent. Any inheritable feature of an organism is called the character, and its two alternative forms are called trait. Like color of the eye can be brown or blue. So character is color of the eye, trait may be brown or blue. Hair texture may be curly or straight. So hair shape or texture is what? Character, and curly or straight are the traits. So mark the definition of character and trait. There are a lot of terms here. Now Gregor Johann Mendel is known as father of genetics. Underline his name. He was actually an Austrian monk, and in his monk he studied the pea plant and he deduced the laws of genetics. Just like we have Newton's laws of motion, there are first, second and third laws of Mendel's law of inheritance. So just underline Gregor Mendel, who was an Austrian monk, and he is known as father of genetics, and he proposed Mendel's laws of inheritance, about which we'll study later. In this modern applications of genetics, mark the term genetic engineering. Genetic engineering is the technique in which genetic constitution of an organism is altered by introducing new genes in the chromosomes. Now, with increasing cases of diabetes, scientists found it very challenging how to produce insulin. So, what did they do? They changed the DNA of bacteria, because bacteria was known to have the fastest rate of multiplication. So they genetically modified its DNA and introduced an insulin producing gene in the DNA of bacteria. And when the bacteria now multiplied, this insulin producing gene also multiplied with it, and these bacteria could be turned into insulin producing factories. So this is how genetic engineering is done. You alter the genome or the genetic constitution of an organism, and you make it produce something which is beneficial or useful. So bacteria find great use in genetic engineering, especially in insulin production. Next comes genetic counseling, in which the newly married couples are counseled by the specialists, like if one of the parent has any trait which can be genetically transmitted to the child, what precautions they need to take, or beforehand only their gametes are genetically modified so that the child does not inherit the diseases. There are some diseases like hemophilia. These are transmitted by defective genes. Hemophilia, known as bleeder's disease, in which the blood of the patient or person does not clot for long hours. Often the person might bleed to death. A small cut or injury or prick by the needle can be a cause of the death. That is hemophilia or bleeder's disease. Thalassemia, sickle cell anemia. And thalassemia, your blood cells, RBCs, get destroyed on their own. So the hemoglobin count always remains low in such patients. Sickle cell anemia, the RBC, which is supposed to be biconcave disc-like, it's supposed to be like this, having a depression here, is like a sickle, the sickle with which you cut the grass. So this is sickle. So since they become D-shaped, these oxygen carrying ability of the RBCs decreases, hemoglobin content is less, so person is anemic. So this is sickle cell anemia. So underline the name of three disorders. They are mainly related to your blood or your circulatory system: hemophilia, thalassemia, sickle cell anemia. Here defective hemoglobin is there. They are all genetic diseases which can be transmitted from parents to children. So such parents need to take caution before they plan a family, that what if the child also inherits these diseases. So they are first subjected to genetic counseling. So mark for what is genetic counseling and what is genetic engineering. Now we'll move on to chromosomes. We have this previous knowledge that chromosomes are the carriers of the units of heredity, because chromosomes bear genes. Now scientists did what? They fitted cameras inside the microscope, and from the lens of the microscope pictures were taken. You know that we have 23 pairs of chromosomes. These are the pictures of 23 pairs of chromosomes. These 22 pairs are known as autosomes, and this one pair in female, that is XX, or in males, that is XY, is known as sex chromosome. So overall we have 23 pairs of chromosomes, in which 22 pairs are autosomes and one pair is sex chromosome. So their pictures were taken individually of the chromosomes. They were paired in the pictures. Their pictures were pasted on a chart, and they are paired in their pictures. Similar autosomes are kept together. They are numbered. Sex chromosomes are kept separately at the end of this table, and this kind of depiction is known as a karyotype. So what is a karyotype? The human chromosomes arranged in pairs in order of their size and shape, ranging from the largest to the smallest, followed by the sex chromosome. This depiction or this chart is known as karyotype. So tick the word term karyotype, and this is the picture of this is what a karyotype looks like. Now coming to the differences between autosomes and sex chromosomes, which is always asked. So first difference would be autosomes. They are 44 in number, or 22 pairs of autosomes are there. And for sex chromosomes, you will write they are 2 in number. They are 2 in number. Second would be autosomes are always, every members of each pair are similar in autosomes. They are 44 in number and members of each pair are similar to each other. But in sex chromosomes, if it is a female, the members of the pair would be similar, that is XX type. But if it is in males, the sex chromosomes, the members would be dissimilar, partners would be there like X and Y. So autosomes 44 in number, sex chromosomes 2 in number. Autosomes, members of each pair are always similar to each other. Sex chromosomes, members of each pair are similar in females, XX type, and dissimilar in males, XY type. Third, an important difference is sex chromosomes help to determine the gender or sex of an individual. They determine the gender or sex of the individual. If it is XX type, it would a girl child would be born. If it is XY type, a boy child would be born. So they determine the gender of the individual. Autosomes control all other features besides gender determination. They govern all other features or control inheritance of all other features besides gender determination or besides sex determination. So these are the 3 differences between autosomes and sex chromosomes. Overall, we human beings have 23 pairs of chromosomes, in which 22 pairs are autosomes and 1 pair is sex chromosome. You have an idea here about the number of chromosomes in other organisms as well. You are not supposed to learn, but this is just to give you an idea. You could learn one from the plant, like onion or maize can be learned, one from an animal. Lion and tiger, their chromosome number happens to be the same, but genes would be different, remember. So you could learn one example from plant and one from animal, just for giving as an example. Homologous chromosomes has been explained to you earlier. A pair of corresponding chromosomes of the same shape and size, one obtained from each parent, or in other words, one maternal and the other paternal. A pair of chromosomes of the same shape and size, one maternal and the other paternal, add there, bearing the same gene at the same location. Bearing the same genes at the same location. Next, we'll move on to sex determination, son or daughter. Just now it was explained to you that if it is XX type, it is a girl, and if it is XY type, it is a boy. But who determines the gender or the sex of the individual? Now, it is a common myth or a disbelief or ignorance in the society. They believe that it is the mother who determines the gender of the child, because mother gives birth to the child. But it is not so. Actually, it is the father who determines whether the child born is a son or a daughter. So underline, the sex of the child depends upon the kind of sperm which fertilizes with the egg, because sperms are of 2 kinds. They can be X type or Y type. So 2 kinds of male gametes are produced by the testes, either X type or Y type, whereas only 1 kind of gamete is produced by the female or the ova, X type. So female is producing only X type, male is producing X or Y type, and it is just a matter of chance whether X type of sperm fuses with the ova, which is only of X type, to form a daughter, which is XX, or Y type of sperm fuses with the ova to form XY, that is a son. So it is the sperms which are of 2 kinds, X and Y type, but ova is only of X type. So the gender or the sex of the child is determined by the father and not by the mother. It comes in the exam and you have to write it in this way, that if X type of sperm fuses with the ova, a daughter is produced. If Y type of sperm fuses with the ova, a son is produced, because sperms are of 2 kinds, X type or Y type, but ova is only of 1 kind.